Good morning, everyone. This is Shelley from Zhang, leading exploratory biology team at HDB Wuxi AppTech. Today it's my pleasure to introduce to you the comprehensive target discovery and validation platform at HDB Wuxi AppTech. So we have an AGM, short for Advanced Genomic Manipulation Platform, that's built around CRISPR technology. And we provide comprehensive phenotypic functional and assay solutions encompassing wide choices disease models. We also have over 1,000 cancer cell line banks that can be utilized for target cells options. We also have mouse genetics and in vivo gene editing platforms. So all this give you integrated platform to support your novel target discovery and validation. So our HDB Advanced Gene Manipulation AGM platforms is having all kinds of innovative technologies, know-how, and multiple delivery systems for gene manipulation, including also comprehensive options from cellular, tissue to in vivo. It provides valuable tools and solutions for in-depth mechanistic studies and proof of concept evaluation. Our CRISPR-centered AGM platform can give you options, solutions for multiple cell line generation, solutions including knockout, knock-in, overexpression, knockdown, and particularly for overexpression knockdown, it can be constitutive or DOX-inducible. We also have a guide library screen as just covered by Dr. Wang. So this can help you identify synthetic lethality genes, drug modifier for sensitivity resistance, and also E3 ligase. So these are for molecular glue mechanisms. We have also extended our CRISPR technology, our AGM, to primary cells, including primary immune cells, as well as iPSCs. And CRISPR technology has also been upgraded to base editing and prime editing for better precision and versatility. We provide comprehensive phenotypic assays and models, including just, for example, here, high content and IQ, and also PROTAC target protein degradation platforms, including high-bit knock-in, DTAG, and smash knock-in. So all these leading edge capabilities can support target discovery, validation, and new modalities. So the library screen is for novel target discovery, such as genes for synthetic lethality or drug modifiers, which has just been comprehensively elaborated by Dr. Wang. And this is just a case study showing the drug-resistant sensitive target hits validated by knockout study. So for our CRISPR, we can do multiple target knockout. So this is an example showing a single or double target knockout in cancer cells. And it's actually, you can do 3 target knockout in either sequential manner or simultaneously, depending on the crosstalk of these targets. The CRISPR-Cas9 technology proved to be efficient, effective approach, particular for multi-target manipulation. And we can also perform CRISPR-based knock-in to validate drug resistance studies. and also sensitivities. So this is just to show that CRISPR knocking of mutation can produce a IC50 shift as demonstrated here. So this just validates these mutations functioning in drug sensitivity or resistance. And CRISPR base editing is an upgraded technology of conventional CRISPR, and it provides more precise genetic manipulation on a single base pair cases. And here it's showing a high percentage mutation for A to G or C to T confirmed by NGS. And CRISPR prime editing as a more advanced technology, it enables higher precision, efficiency, and versatility. And here is the example showing that we have successfully inserted his tag into endogenous HAG3 locus by prime editing. Again, this is standard sequencing results. And we provide a comprehensive assay format and technologies to support target validation. You can see it encompasses biochemical assays, radiometric assays, cell-based target assays, so such as reporter assays, and also Western blots using ECI as well as LIQUOL. And we also have JAS that has a more quantitative for smaller amount of samples. And also we provide high content multiplexing assays in a quantitative high throughput mode. Phenotypic functional assays for primary cells and as well as engineered cell lines, and including various technical platforms and multiple therapeutic focuses. Also biochemical assays and mass spec. And we also have multimodal plate readers, just listed here. And one example, Luminex 3D, up to 500 multiplexed barcoding readings, MSD and LIQUOL Odyssey. So for all these multiple solutions, as well as liquid handling systems as listed here. At HDB, we have 1,000 tumor cell bank with extensive cell type coverage, including 18 different tumor disease models and very wide geographic diversity, and also provide internal genomic data for over 300 cell lines. We also provide complicated, complex assay platform solutions that's listed here. We have successfully extended CRISPR platform to target manipulation in very tough non-proliferating primary immune cells, including mouse and human primary T cells, human monocytes, dendritic cells, M1, M2 macrophages. And these CRISPR-based knockout of novel targets have been validated by FACS or standard sequencing. So the efficiency is over 50%. And the short time window enables multiple downstream functional studies like RT-PCR ELISA for cytokines in different primary immune cells. So this just opens up horizon and explorations in the very tough field immunology. So we can interrogate novel targets in immune cells. At HDB, we also provide comprehensive in vitro immunoassay platforms, including immune cell activation for multiple kinds of immune cells, PBMC T cells, NK, monocytes, etc., and immune cell co-culture assays like mixed lymphocyte reactions for assessing T cell activation, M2 T cell co-culture, MDSC T cell co-culture, Treg T cell co-culture for evaluating different kind of immune cells functionally, as well as immune and tumor cell co-cultures for novel target discovery and validations, as well as immune cell polarizations, so like biomarker profilings and phagocytosis for macrophages. We have developed PROTAC platform at HTB, so this has been extensively utilized, applied for multiple novel targets across multiple kinds of cancer cells. And we also extended this PROTAC platform to primary immune cells. We have very short turnover time, around 1 week, ensures sufficient time for downstream functional assessment. So this is very advantageous for non-proliferating primary immune cells. And the high delivery efficiency ensures excellent assay window, over 100-fold, in the case of step 3. And this in the pooled primary cells shows stability up to 20 days. And this provides options for 384 well-played HTS capability and providing solutions for novel and druggable targets in tough primary immune cells. So here, example of knock-in hybrid in step 3, well-established targets in primary T cells and its functions in inflammation diseases. And we have used a reference compound of PROTAC and showing a degradation step 3 in primary T cells with IC50 well correlated to reports. So this is showing, this PROTAC capability in primary immune cells, helping novel target validation and also screening capability in primary immune cells. We also developed TCR-T CAR-T new modality platforms and demonstrated over 90% high efficiency delivery. So this is advantages, for the downstream processing from the clinical point of view. And TCR-T CAR-T functional validation is performed by co-culturing with MHC or surface antigen overexpressing target cells, followed by cytokine ELISA detections, as you can see the dose curve shown here. We also have developed new modalities such as antibody-oligonucleotides, AOC. This is including rational strategic assay design to efficiently validate siRNA targets and surface receptors for siRNA antibody or ligand conjugates by parallel target cells versus control cells, mRNA, as well as protein expression profiling. We have done comprehensive validations, including multiple layers of optimizations to ensure very high efficient siRNA screenings by RT-PCR as well as ICW. And rational workflow design for target cell surface receptor determination by RT-PCR followed by FACS confirmation for receptor target surface localization. So this is just showcasing our comprehensive siRNA screening assay optimizations, including transfection reagent, knockdown efficiency, comparison of multiple transfection efficiency reagent to select the most efficient and with the minimized cytotox effect, and also transfection reagent volume time point determination, and housekeeping gene optimization for multiple housekeeping genes across different cell lines. Also, different lysis kit validation by RT-PCR to select the best options that has the best knockdown efficiency. So all these comprehensive validation led to very efficient screening of assays. Both the primary screens with 2 dose points as shown here, and followed by the potent hits confirmation for IC50 assessment by a full dose curve assessment. And also parallel with in-cell Western validation in 384 format by LICOR for high throughput assay screening or compound screening. So this is just showing the assay screening. You can see the dose response very nicely, and quantitatively, you can also assess IC50s. So for the target cell surface receptor determination, this is just showing the mRNA expression profiling across multiple targets as well as control cells, so we can determine the most promising candidates to confirm by facts. So this is very important for the conjugation by the antibody. Besides AOC, we also have established ASO platform. So this is carried out by the first step setup and optimization for ASO screening by RT-PCR. So including genomic versus transfection approach and time point determination, etc. in human mouse cells, and ASO screening and hit confirmation in both human and mouse cells within parallel cytotox evaluation. And the hits were further confirmed functionally by RT-PCR Western ICW with functional validation in human mouse primary cells for enzymatic and cytokine studies. We also have set up new tech development for barcoding our cancer cell panel. So this, we can have pooled 10 to 20 cell lines for drug screening together to greatly boost the drug screening efficiency. And it's also applicable for tumor heterogeneity studies. We expand the CRISPR barcoding to 300 to 400 frequently used cancer cell lines with 2 compound validation. So HDB growing comprehensive target discovery validation capabilities propelled by various new technologies provide solutions for drug R&D across multiple disease areas, as well as new modalities, including PROTAC, TBD, CAR-T, TCR-T, AOC, ASO oligos. And also we have technologies and platform for tumor cell panel barcoding and base editing, prime editing, and CRISPR extended to primary immune cells, iPSCs. So overall, all these provide an integrated, comprehensive platform to help provide solutions for target discovery and validation. And here is the contact information for myself. You can email me for any of your science-related questions, or email our business contact Dave for any business-related queries. Again, not to emphasize enough, our mission is to improve health, making difference. Together we deliver. Thank you very much, and welcome your questions in the following sessions. Thank you.